Thanks very much to the organisers and for everyone staying till the end of the day. I hope this is going to be a really nice clinical example of bringing together a lot of what's been covered today, lots of the methods, including the deep learning that Emma presented, for a real clinical case. So, and lots of the coding that has been done in this project has been by the four people here: Mathilde, Hannah, myself, and Sophie, who sadly couldn't be here to present. No conflicts. So I'm going to start with a case example of an 8-year-old girl who's referred to Great Ormond Street Hospital, which is a pediatric hospital in London, with drug-resistant epilepsy. She has an MRI scan, and no abnormality is found by the radiologists. And so it's not clear what's causing the seizures and what could be done to treat it. Drug-resistant epilepsy is not rare. In around 30% of people with epilepsy, the medication doesn't work. But if you can find a focal brain abnormality, you can operate, and this can cure the seizures if this MRI lesion can be found. And so our work on the multi-center epilepsy lesion detection project has been to build the imaging data sets and the AI tools to better understand, diagnose, and target treatment in focal epilepsy. And when we're talking about MRI negative epilepsy, the ones where you can't really see the lesion, in both adults and children, so on the left is from the Cleveland Clinic in America, and on the right is from Great Ormond Street, looking at the cases that were MRI negative, the most common pathology is focal cortical dysplasia. So this is the pathology we worked on first. So in the talk, I'm going to kind of explain what focal cortical dysplasia detection is all about. There is then an interactive Colab notebook. So if you're planning to type along, the short URL is probably the easiest thing to type into your laptop, so I encourage you to do that. We'll be going through that together. And then at the end, talking about how we've been working to translate this clinically. So I'll just leave that on for another 10 seconds in case anyone wants to catch that URL, and then I'll move on. Okay. So focal cortical dysplasias are a malformation of cortical development. That means they start to develop in the fetus, but seizures don't tend to start until early childhood. And they're kind of notorious for having these very subtle imaging features. But what radiologists do describe are, and I've taken you to the right section on the right of a T1 and a flare image with an FCD in it, so you can see if you can spot these features. Abnormal cortical thickness, blurring of the gray-white matter boundary, flare or T2 intensity changes, and the kind of classic sign is this transmantle sign, which is a hyperintensity in the flare going from the lesion down to the ventricles. Abnormalities in folding pattern, and then often you're looking at asymmetries of any of these features. So we will hopefully do a poll. Is this going to work? Otherwise, we'll just do by hands. No, okay, we're going to do by hands. Okay, so I've got some boxes there. We've got A, B, C, D, E, F, G. So I'm going to just go slowly through the letters, and I want hands for where you think the lesion is. I'll give you another few seconds to have a think. So hands up for A. Hands up for B. Hands up for C. Hands up for D. Hands up for E. F. And G. Okay, thank you. That was very good. I think I was kind of even distribution across, except a slight peak, I think, came for B, which is really good. Perhaps there's a couple of radiologists here. So if you look at B, you can see some blurring and you can see the hyperintensity of the white matter I talked about. Look at box B, and then going into the ventricles, you can see a white streak. That's the transmantle sign that often people are looking for. But I think one key thing is, now that I've pointed it out to you, hopefully a few other people can start to see what I'm talking about. And this is really where AI can help, because if it can point to a potential abnormality, you can go back and look at it and say, yes, I think that does look like an FCD, or no, that's some kind of imaging artifact. Okay, so no polls. So together with 22 hospitals around the world, epilepsy centers, we created a surface-based imaging data set, which had over 600 patients with FCD and 400 controls. And in the first version of this was really surface-based for the privacy reasons that were discussed earlier. Each site could process their data locally, extract features to the surface, and then just send that without real problems of anonymization, and it was easier in terms of the ethics. This is both pediatric and adult data, and what we received are those surface-based features and manually delineated lesion masks also mapped to the surface. So we know the characteristics of the features on the surface and where is lesion and where isn't lesion. So the first thing we did is we mapped the lesions to a symmetrical template brain. This is the FS average sim template. Because in theory, these dysplasias can occur anywhere in the cortex, but actually the first time you've put together a data set like this, you start to see that they do occur everywhere, but the blue areas is less frequent and the red areas are more frequent. So the temporal pole and the superior frontal sulcus are real hotspots. From a radiologist's point of view, this could be useful because it could guide where you should be looking more closely. But biologically, it's also interesting, kind of from the kind of multi-scale biology, why are lesions occurring in certain brain areas and not so much in other areas? And I think this is a really unsolved problem. So this is the overview of the pipeline. I'm going to zoom in. So firstly, we have this local site feature extraction, which I'll just go through now. So using these surface-based methods that we've been hearing about today, we decided to extract all these features that radiologists use. So cortical thickness, the distance between the white and gray surfaces. The blurring of the gray-white matter boundary in T1 is sampling the values within the cortex and within the white matter and computing that contrast. So when the contrast is low, that blurring measure goes down. You can also sample the flare volume at different depths and into the white matter to try and capture that transmantle sign. And then you can also look at folding patterns of curvature, sulc, and intrinsic curvature. And finally, because we're using a symmetric template surface, you can just subtract the two to get the asymmetries of any of these features as well. Then we do a number of harmonization and normalization steps. So this is multi-center data, different scanners, different sequences. And so when you take a feature like cortical thickness, calculated on the surface, and you average it within each subject, we're plotting against age to give a kind of biological feature. You're expecting to see a roughly linear decrease with age. But what you really see is a site effect. So the orange site, each site is a different color. The orange site has lower cortical thickness. The green site has much higher. And so we use harmonization, combat, to remove this site-based effect. And once that's removed, you can see this stronger age effect. We then do a number of normalization steps. So firstly, we do intra-subject Z-scoring. This helps to remove this age effect, sex effect, and general intra-subject variability in measures like thickness. We can do the asymmetry, which is kind of within subject normalization, because what's too thick on one, generally the brain is relatively symmetric, and so if something is abnormally thick, it should come up with the asymmetry. And then we also do normalization by controls. So the front of the cortex is thick, the occipital cortex is thin. We've seen that already today from Matthew's atlas. And so what is thick is also relative to the area that it's in, so we do Z-scoring by the corresponding vertex in all the controls to kind of highlight what's abnormal. We can have a look at those features within the lesions. So this is a UMAP where each dot is the average, is the features for the lesion mask, and dots that are closer together have more similar features. So it's a kind of dimensionality reduction technique. And here they've all been colored by the features. So you can see this intrinsic curvature as a folding measure is pretty abnormal for all the patients. Cortical thickness tends to be a bit increased. Maybe there's a group at the bottom that are decreased. Gray-white matter contrast is consistently low. Sulcal depth is not potentially the most informative feature. And then the flare features, when we had it, generally hypo-intense in the cortex and either hypo or hyper-intense in the white matter. So the top right cluster there, if I've got a laser there, that looks like the transmantle sign group there. So I guess one thing is the features are abnormal in the lesions, but also they're pretty heterogeneous. So you can't just threshold and say this is going to be lesional because it's a bit more complex than that. So, excuse me, this is where the machine learning starts to come in. We have an unfortunate position to be able to have a train and test cohort and also some independent test sites to kind of evaluate how it's generalizing to other sites. In our first iteration, it was a multi-layer perception. So just looking at each vertex in isolation, having the pattern of features and knowing whether this is lesion or non-lesion, and just trying to learn patterns of features that correspond to lesion or non-lesion. This is the first paper that we did on this data set. But we have since kind of moved on to the kind of more complicated graph convolutional approaches that Emma was talking about. And so rather than taking each point in the surface in isolation, you can start incorporating contextual information, the neighbors' neighbors, and building into these more complicated UNet architectures. And so this is an overview of the architecture. You have input features on the top left, so that's all those raw features, the normalized features, all go in. We have 32 features going in. You have this downsampling and upsampling operation. So you go from the high 160,000 vertex sphere, downsampling it till you go down to a 42 vertex sphere at the bottom, and then upsampling again to get that anatomical precision back. We have a number of additional tasks. So we have segmentation, which is on the top. Because this is a really difficult lesion to mask, we have some extra things to help deal with that uncertainty. So we have a distance regression task, which says how far are you from the lesion? And this penalizes predictions that are very far from the lesion, but not ones that are directly next to the lesion mask, which could account for kind of if the mask isn't exactly perfect, you're not penalizing those predictions so badly. And then for when the mask is completely wrong, we just say, is there a lesion somewhere on the hemisphere? And then it can still learn something from that task as well. And that's the hemisphere classification task. Having a comparison of those two methods, the pervertex, which is the multi-layer perceptron, and the graph convolutional approach, you can see that the perceptron is picking up the lesions. The red predictions are within this black line, but in each case there's a false positive here, and some false positive clusters here, and here, and here, whereas the graph convolutional network is consistently picking up the lesion with a much nicer prediction. And when you look across the whole cohort, the kind of issue of false positive drops off a cliff once you start incorporating this whole brain context, and it can start to decide this is the cluster I think is actually the lesion. Kind of in terms of statistics, the one that really jumps up is the positive predictor value. If there's a prediction, the likelihood of it being a cluster is now 70%, whereas before it was 40%. And then this is wrapped up into a new subject prediction pipeline that will go from end to end. You put in the T1 and flare images. If you have them, it will run through the free surfer extraction, do all the harmonization, normalization, run it through the network, and then produce a prediction report, which you can then map back to your original MRI and review the prediction against what you saw originally. And this is two prediction reports, but I'm now just going to switch on to the interactive collab, because actually one of those patients is in there, so can we switch? And for those who haven't, so this is what you may or may not have up on your computer. The first cell takes about a minute to run, so I think I've already pre-run it, but beyond that, it should run exactly straight out of the box. It's got two example subjects uploaded. So the first one, so if you run the first cell, and while it's running, we can have a look. The first is patient 0. It's an 18-year-old male with focal epilepsy. The seizure is not controlled by medication, and they were investigated for possible epilepsy surgery. And as part of this investigation, they had an MRI scan. So we're first going to load in the T1 and flare volumes. They've been co-registered, so can we run the next cell? Yep, and then the one below. So we've got a little viewer. If you scroll down a bit, it should be an interactive viewer. So you can slide the slice index just at the top there, Logan. And then you let go and drop it. And so if we go at 30, 60, you just have a quick through. And is anyone, if you put your hands up if you've also got this up on a laptop. Otherwise, we'll just do it here. Okay, no one else is not on their laptop, but you can if you want to. Can you do 30, 70, 100, 150? 30, that's 30. One of these slices has a lesion in, so that's why I'm just wanted to see if you can spot any of the features we've talked about. And then we're going to have a poll. 1 is, I've got no idea. 2 is, maybe I can think where it is. And 3 is, I'm pretty sure I know where this lesion is. And then 150, yeah. Okay, so hands up 1 if you've got no idea where this lesion is at the moment. That's most of the people in the room. Good, good. Means there's ways to improve. 2 if you've got a pretty good idea of where you think the lesion is. Okay, and then 3 if you don't need anything else. I know where it is. Great. So everyone could improve their confidence if we keep going down. If we scroll down now, we've just done that poll. Let's load in. So we're now going to, you could in theory load up all those features. We're just going to focus on 3 for now: thickness, blurring, and gray matter flare. We're going to start with the right hemisphere. That's a clue, it is actually on the right hemisphere. And then we're going to plot those features without any pre-processing, just the raw features onto the surface. This has got it's going to load in those 3 features and then plot them onto the mesh surface for the right hemisphere. Takes about 5 seconds to do. Okay, so that's cortical thickness, gray matter contrast, and flare at the bottom. if we just hold on there for a second, let everyone have a look at that. This is the raw feature. It's not been processed. I guess one thing you can already see is there's variability across this surface. Hard to tell necessarily which variability is normal and what might be lesional. But having seen these plots, we're going to do that 1, 2, 3 poll. So 1 is still no idea. 2 is I have a vague idea, and 3 is confident. So 1, still no idea. So a couple of people have changed now to 2. I think I've got a vague idea. A few more people. And 3, I'm pretty confident now I know where the lesion is. And we got 1 or 2 3s now. Great. Let's keep going on. Okay, so let's just stop there. So we've talked that cortical thickness varies as a function of age. So we Z-score within subjects to remove that variability. But then there's also this stereotype pattern across individuals. So if we load this cell, this is just average across all our controls. After that intra-subject Z-scoring, you can see that kind of the central sulcus is very thin, the occipital cortex is very thin, but other areas are expected to be thicker. So the frontal cortex, and I'll zoom you actually, if you have a quick look there, this area you would normally expect to be quite thick as well. This area would also expect to be quite thick. You have to kind of think about these things. So we then remove this interregional variability by normalizing by these maps for each of those features. So we're now going to plot those normalized feature maps. Hopefully things start to become a bit more clear, but maybe not perfectly clear. Got 10 seconds of this. And the other thing I'm not plotting here is the asymmetry plots that we also do to kind of highlight that. So that's cortical thickness after the normalization, that's the gray matter contrast, and that's the flare. So this is going to be the third of 4 polls. The third one, we're going to go 1, 2, 3 again. So 1, still no idea. Okay, 2, not sure, but maybe I got an idea. Most people in the room now. And then 3, I'm pretty sure I know where it is now. Okay, so we've got about 4 or 5 of those, so great. Let's keep going down. Okay, so at this point, we go to the AI. It sees 300 or 400 examples of what these lesions look like, learns the thresholds, learns the patterns of what these lesions look like. Just quickly for augmentations, examples that we use in this training, we have kind of the same things you might have seen on 2D image augmentations, so brightness, contrast, gamma, noise. These are all intensity-based augmentations applied to the features. And then we have spatial augmentations, so flipping, spinning, and then nonlinear warping of the tissue, to augment the amount of data, improve generalizability of the trained algorithm. So we've trained our algorithm. We're going to put those normalized features through this algorithm and have a look at the prediction. If we keep going down, we should see the prediction. So is this kind of tallying where people were thinking this is where the AI thinks the lesion is? General nods. This is where you started to think, yeah. So this is an area that was, I think, thinner than expected. It was, in terms of its contrast, it was definitely reduced contrast, and the gray matter flare was hypo-intense. And then, if we keep going down, this is the interpretable report for this subject. So this is what's then fed back to the radiologist or whoever wanted to look at it. It says, this is where I think it is on the surface. It maps it back to the MRI. This is the patch that I think it is. Here on the right hemisphere is the kind of left-right flip that radiologists like. And then kind of a crucial part of this is interpretability. So it says what the features were inside the cluster, but also how salient each feature was in making this decision. So the color is the saliency. So for this lesion, reduced gray matter contrast, increased cortical thickness, my mistake, and flare hyperintensity in the gray matter were the things that really drove this decision. And then we can go back to our MRI scan and look at T1 and flare. And actually, at this point, can we scroll all the way back to the top to the viewer? And then if you were looking blind, just there. And if we go to slice 70, which is the equivalent slice, roughly 70 is fine. Yeah, so we're now, can you see that blurring here? And maybe that looks like a T1 now. Why does that look like a T1? Anyway, on the T1, you can see this hypo-intensity here and potentially weird thickness. And that's the kind of very subtle, but once it's been pointed out, you can start to see those features. If anyone wants to have a go, there's another subject 01, that was actually MRI negative and is really hard to see. Even when I'm told I can't see where it is, but you can kind of do the same thing, go through the normalized features and see where the classifier predicts the classifier is right for this example, so kind of you can use that to see if it can help you find the lesion. Okay, can we go back to the slides? Cool. So in the last 10 minutes, I'm just going to go through how we've been working to translate this clinically. Oh, we do have this anyway. Okay. So the three kind of ways we've been working with different people to translate this. The first is with neuroradiology, second is with the neurosurgeons, and then the third is to kind of help sites around the world use the tool. So going back to that 8-year-old girl that we met at the beginning, drug-resistant epilepsy, the MRI was reviewed and nothing was found. The radiologists at Great Ormond Street can now request that it gets run through the MELD pipeline, the MELD protocol, and then it returns this report for that child. And there's a predicted cluster in the insula where in this patch here you can see blurring of the grower matter boundary and increased, there's kind of an abnormal folding pattern. And the radiologist could then go back to the MRI scan and say, yes, actually, this did look like an FCD. This was confirmed by them, and then this child is now able to proceed to surgery. And actually, when you go back to this flare image in the same area, there is a bit of a weird flare. It's kind of, maybe my mouse is hiding it, but there is something there that actually, once it's been pointed out, is a bit easier to see. The second way we've been working is with the neurosurgeons. So when they really can't see a lesion, often they will then implant electrodes into different parts of the brain to try and measure the seizure activity directly, and then they could then resect wherever the seizures are coming from. And in this clinical trial, if they made their plan and MELD was then run, if there was a cluster that they weren't going to implant, they would then add an additional electrode into that cluster and check whether the seizures were coming from that patient. This was a COVID trial, so we ended up with quite a few patients, but of the 10 that were considered possibly implantable, 4 had FCDs. And in this one particular example, this is a cluster that was a new electrode that was introduced because of the MELD algorithm. It was part of the seizure onset zone. They changed the resection plan to include this patch of tissue, and the patient has been seizure-free since. So at least in one really nice case example, it had a direct impact and good outcome as far as we know. And then the final thing we've been doing is training people. So we have this code, it's all openly available to use. The pipeline runs as a single command line, so you have your T1, your flare, it'll run free surfer, extract all those features, do all the normalization, calculate the harmonization, asymmetries, into subject normalization, run it through the classifier. This is the version with the multi-layer perceptron, but we've also just released the graph convolutional network, which then produces this prediction report, which is then mapped back and can be reviewed. And we've trained with epileptologists through the International League Against Epilepsy and a European-wide reference network. So we now know of about 65 scanners where people have been kind of testing this out on their patient cohorts as well. Just to kind of pick up on Matt's point from earlier, this is not a clinically licensed tool. It's only really used in a kind of research framework. And just in the last couple of minutes, so MRI negative epilepsy, FCD is the most common cause, but actually it makes up less than half of the cases. And there's a whole range of other pathologies in this other section that we have to be thinking about. And so kind of our new MELD focal epilepsies project is to collect many of these pathologies. So not just FCD, but HS, gliosis, these other kinds of tumors that can cause seizures and are sometimes missed. And we've been doing the data collection for this. It's still ongoing, so if you have access to these lesional patients and you'd like to be involved, I think we have 20 sites who've contributed data so far, but we're still always keen to receive more data. And the aim here is where in the brain is causing the seizures, where in the brain's causing the seizures, what is the specific abnormality that's causing them. We also have the post-op scans this time, so we can say where should be operated or where was operated and what was the outcome. And that kind of will help us to predict, given the lesion, given the location, and given a particular operation strategy, how likely is this patient to become seizure-free. So just to summarize, we talked about FCD and the features, the kind of surface-based features that can be used to characterize it and then detect it. And we have these open surface-based tools for this automated lesion detection that we try to make as interpretable as possible. And we've also now been working with the clinicians to kind of facilitate the translation of these kinds of tools into clinical practice. And so with that, I've got loads of people to thank because this project happens with the contributions from sites all the way around the world. I'd like to particularly thank Mathilde, Sophie, and Hannah, who did lots of the coding with me on this, and our funders. And we have just got some funding to expand, so if you like the idea of working on this clinically translational epilepsy problems, please get in touch. Thank you very much.
So we're going to wrap this session up by doing a panel session. So if I could invite all of the speakers from this afternoon onto the stage. I know Emma isn't here.